So thanks to the organizers for the invitation. It's a wonderful city, and it's also quite spectacular for a Norwegian to go someplace which is colder than at home. Now, the title of my talk is actually very misleading, because in the end I'm not going to show you anything, or hardly anything, on metabolism. So I think a more correct title would be Cardioprotection by Connective Tissue Growth Factor in Myocardial Ischemia and Heart Failure. And I would like to emphasize that the main drive behind the data I will show you is not from my own lab, but from the laboratory of Professor Håvard Attramadal, also at the University of Oslo. The previous speakers have reminded you of that heart failure is due to changes following an index event, which may be acute myocardial ischemia, pressure overload, volume overload, or pregnancy, as Denise talked about, leading to changes in the heart muscle itself in the attempt to compensate the different demands of work. And those changes are accompanied by damage which may lead to symptoms in the heart many years later. And there are sort of three basic mechanisms underlying the heart's adaptation to injury. One thing is hypertrophy of the heart muscle, its repair of the injured muscle, and its cellular protective mechanisms. And these mechanisms work in homeostasis, trying to maintain a normal heart muscle function. We have specifically looked at connective tissue growth factor. That belongs to a family of growth factors which are expressed during embryogenesis, but which is then repressed in the postnatal heart. And we find then that in the failing heart, we have a large increase of cardiac expression of CTGF, shown both at the protein level in the pig hearts and shown as RNA level in human heart failure, especially of coronary artery disease background. Connective tissue growth factor is in the literature difficult to understand what it does in the heart. It is connected with myocardial fibrosis. The problem is we don't know what the receptor for CTGF is, so we have no pharmacological means to address its function. If one makes knockout mice of CTGF, they die during fetal life anyway. This is not due to malformations of the heart, but probably due to disturbances in the conductive system. So our strategy to try to understand what does CTGF do, which I may add, if you find the factor increased in a failing heart, it can be something which contributes to the pathophysiology of heart failure development. It can be something which is due to a compensation to a beneficial factor to try to contradict the injury, or it could be something which is completely without any importance. So what we did was to make mice with a cardiac-restricted overexpression of CTGF. And CTGF was then added to an alpha-MHC promoter, which is expressed then only in the cardiomyocytes in themselves. And when we looked at heart tissue of the transgenic animals, we found an approximately 70-fold increased expression of CTGF protein. And there is a small endogenous expression in the non-normal littermate controls, hereafter abbreviated NLC throughout all the slides. And the same, the densitometric analysis shows that this increase is large, and also we can see that the protein, the increased CTGF, is in the cardiomyocytes in themselves. We then did just the phenotyping of the unstimulated CTGF mouse. And the main finding is really that the heart weight is reduced in these mice, and the heart weight-body weight ratio is induced. So the mice are slightly smaller in themselves, not actually visible in the body weight shown here, and the heart is smaller. The function, as evaluated through pressure-volume loop analysis, really confirms that the function is normal in almost any endpoints, but the left ventricular end diastolic volume is reduced in these mice, which probably just reflects that their hearts are smaller. But they tend to sort of have everything a little bit less than the legitimate controls. I said some slides ago that what is known from the literature is that there seems to be an association between CTGF and fibrosis in the heart. So we try to look first at the fibrosis in the transgenic mice and find then that the transgenic, as opposed to the heterozygous, have an increased expression of mRNA encoding for procollagen 1 alpha and 1 beta, as well as hydroxyproline. And through a mouse-own trichrome staining, you can see that there is a small but increased fibrosis in the heart muscle in itself. And we also did a staining to evaluate capillary density with an anti-CD34 positive antibodies, and we find that there is a small but significant increased capillarization in the hearts of the transgenic animals. This is, however, not accompanied by an increased blood flow, so it's possible that this apparent increased capillarization only reflects that it's the same vascularization but with smaller heart cells in between. So we then want to see what the role of CTGF could be in a heart failure model, and we did the aortic banding of mice, and thereafter 6 and 12 weeks later looked at heart function. And this rather busy table basically shows that while aortic banding leads to an increase of heart weight in shams, let's see, that's here, this was attenuated in mice overexpressing CTGF, and the heart mass, if we calculate it, for instance, in relation to the tibial length, is reduced in the heart, the mice of hearts of CTGF transgenics, as opposed to the wild types, which have then a dilated cardiomyopathy. And also, gene expression of hypertrophic markers were reduced after pressure overload in CTGF overexpressing mice. The expression of ANP was reduced compared with the wild types. Expression of BMP was reduced, and alpha skeletal actin, which is an early marker of hypertrophy, was reduced. And the contractility was better preserved in the transgenics compared to the controls. I think I'll just leave this slide here and not comment on every single endpoint, but just point out that changes occurring in the transbanded wild types are reduced in the banded transgenics. So there is an overall protective effect against hypertrophy in the transgenic mice. We also made heart failure through stimulating the mice with isoprotenolol, and found likewise that the changes induced in the wild types were improved in the hearts of the transgenic mice. So we then wanted to see if CTGF actually contributed to the pathophysiology of ischemia reperfusion injury. And this was done through isolating hearts, hanging them up in the Langendorf system, and inducing global ischemia followed by reperfusion. We could then see that hearts of transgenic animals had an improved recapitulation of left ventricular developed pressure. The depression of end diastolic pressure was less evident, heart rate was unchanged, and of course, most importantly, infarct size was reduced in the transgenic mice. We did see that before induction of ischemia, transgenics had a higher coronary flow. So in theory, the infarct size reduction could be due to a difference in coronary perfusion. So we changed the mode of heart perfusion to a constant flow model. And we're very happy to see that the myocardial protection afforded by the transgenic animals was still valid. So there is a direct anti-ischemic effect. But we do have the possibility that mice which have all throughout their life been transgenic for CTGF in the heart have secondary changes in the heart, which may be the protective factors and not CTGF itself. So in order to address that question, we perfused isolated hearts with human recombinant CTGF given into a sidearm of the aortic cannula 10 minutes before induction of global ischemia and reperfusion. And again, we were very happy to see that recombinant CTGF given acutely attenuated the depression of left ventricular developed pressure, and it could reduce infarct size. So the question is then, why is connective tissue growth factor cardioprotective? And we tried to explore, well, we have tried to explore very many possibilities, but the one possibility I am going to present is, of course, then the most successful one. We wanted to know if the reperfusion injury salvage kinase pathway, which has been described by Derek Yellen and Derek Hausenloy, could be involved in this. And basically, Derek say that during preconditioning, which is a form of adaptation to tolerate ischemia better, you have factors secreted which act on the cardiomyocyte, which will lead to phosphorylation of the PIK kinase pathway. And this, in turn, will phosphorylate AKT, which Denise Hilfiger-Klein also had on her slides as a cardioprotective agent. And one of the downstream effects of AKT phosphorylation is phosphorylation of GSK3 beta. That leads to inactivation of GSK3b. When GSK3b is inactivated, we stop inhibition of glycogen synthase, and one of the end effects of that is an increase of glycogen, which you all know is a substrate for cardiomyocyte metabolism. And this is thought either through glycogen synthase effect or through other downstream effects of GSK3b to lead to cardiomyocyte survival. So we then just wanted to see which factors are activated in the CTGF hearts, and we found an increased phosphorylation of SMAD2, but I will not go into that pathway in this talk. That's not related to the risk pathway in itself. AKT was phosphorylated in the hearts of the transgenics, so was GSK3 beta, and we found then reduced phosphorylation of glycogen synthase, and this was accompanied by an increased cardiac content of glycogen in the transgenic hearts. In order to address if PI3K also had a functional effect and not only led to increased tissue expression, we gave our, no, actually, sorry, I was on the wrong slide here now. We stimulated isolated adult cardiomyocytes with the PIK3 inhibitor LY29 blah blah and stimulated them with CTGF. And in the we found then that CTGF induced phosphorylation of AKT, and this was dose-dependent. And this could be inhibited with the PI3K inhibitor. So there seems to be a link between CTGF acting through PIK3 also functionally. And the same then applies to the downstream factor to AKT. GSK3 beta is less phosphorylated in the presence of LI. Sorry, it's difficult to present data generated by other people, and now I'm making a mess of the sequence. The previous slide was actually then showing that the PI3K inhibitor influenced the signaling of CTGF, and we found the same with an AKT inhibitor. And this then gives evidence to the hypothesis that indeed CTGF acts through PIK3 and AKT, leading to inactivation of GSK3 beta and increased glycogen synthase. So to conclude this, increased tolerance against ischemia reperfusion injury and heart failure were observed in transgenic CTGF hearts. CTGF acts directly on cardiac myocytes and stimulates the AKT GSK3B salvage kinase pathway. And this is a picture of Håvard Attramadal, who is then the one who generated the transgenic mice and who has done most of the experiments. Also in my group at the University of Oslo, we have been engaged in most of all the ischemia reperfusion part of this work. Thank you.